The aim of this experiment is to extract DNA from bananas using easily accessed chemicals that you could even find at home. For this experiment, you will need a ripe banana, knife and chopping board, a balance, pestle and mortar, 2 clear tubes, plastic or glass, but they must have lids, a 1ml syringe, a pipette, funnel and filter paper, a measuring cylinder, either 10 or 20 ml because you are measuring 12 ml with it, salt solution, which is 30 grams per litre, washing up liquid diluted 1 to 5, and cold ethanol, preferably kept in an ice bath to keep it particularly cold. In step 1, you are going to weigh out 10 grams of banana approximately. Now, if you don't have any weighing scales, you could just measure out 2 centimetres of banana. You then need to put this banana into a mortar and smash it up with a pestle. This is to break down the cellulose cell walls to release the cell contents from the banana cells. For step 3, you need to put the mashed banana into one of the tubes and, using the measuring cylinder, add 12 millilitres of salt solution. This salt solution helps the DNA clump together. For step 4, you're going to use the syringe to add 1.5 millilitres of washing up liquid to the banana mix. Then you need to place the lid onto the tube and shake it thoroughly. The washing up liquid dissolves the lipids in the membranes of the cell, and this helps release the DNA into solution. In step 5, you need to fold the filter paper into quarters, then open it up and place it into the funnel. Then put the funnel into the clean tube, and then you need to filter the banana mixture through the filter paper. This removes the banana pulp and the cell debris. Once you've managed to filter approximately 5 millilitres of the filtrate, you then need to remove the funnel, and with a tube at an angle of about 45 degrees, you need to carefully pipette 5 millilitres of cold ethanol down the side of the tube. This forms a layer on top of the banana filtrate. Finally, you need to put the lid onto the tube and gently swirl it a few times. Where the alcohol meets the filtrate, you should see something that looks a little bit like splayed out cotton wool fibres. This is the DNA. It can take a few minutes of swirling. DNA is not soluble in alcohol, and so it precipitates out. And thus, you have extracted DNA from the bananas.